So now the pollen grains has reached here, that is the stigma part, and female gamete is present here inside the ovule, that is present inside the ovary. This is ovary portion. Inside the ovary, ovule is present, and inside ovule this egg cell or ovum is present. Now let us look at the structure of this ovule. Ovule is generally surrounded by two integuments. Integuments means coverings. So two integuments are present here which cover this entire ovule. Only there is one place here that is called micropyle. At this place the ovules are not the integuments are not present. Rest whole is covered by integument. Only at this portion that is called micropyle, integument is not present. Inside this, there are 8 nuclei. 3 are present on this end, that is called micropylar end, because it is towards the micropyle. So 3 nuclei are present here. 3 nuclei are present towards another pole, that is called the chalazal end, right? So, and 2 are present in the center. The 3 that are present towards the micropylar end, out of that 3, 1, the middle one, that is the egg cell, and 2 are called synergids. So it forms one egg cell and 2 synergid cells. The 3 nuclei that are present towards the chalazal end forms 3 antipodal cells. The 2 which are left, these are called polar nuclei that are present in the center. Now what happens when the pollen grain reaches here to the stigma? Stigma secretes some sugary fluids. These pollen grains, they absorb that sugary fluids and swell up. These pollen grains are surrounded by 2 coverings. The outer covering is called exine and the inner covering is called entine. When they swell up, their outer covering exine ruptures and the inner covering entine comes out in the form of tube. You can see this tube. This tube is called pollen tube. So this pollen tube increases in size and reaches inside the ovary. This pollen tube contains 2 male gametes in it. Now because this is entirely covered by integuments, so this pollen tube can only enter the embryo sac through this opening that is called micropyle. So it enters the ovule through this micropylar end. यहाँ से micropyle से enter करती है. Now it has 2 male gametes in it. One of the male gamete fuses with these 2 polar nuclei and forms a structure called endosperm. So endosperm is triploid structure because 3 इसमें fusion है nuclei का. So it is a triploid structure that is called endosperm. This endosperm is a nutritive tissue that provides nutrition to the developing embryo. And the another male gamete fuses with this egg cell and forms zygote. ठीक है. So 2 type of fusions are taking place in the same embryo sac. First fusion क्या है? Male gamete fuses with egg cell to form zygote. Zygote is diploid structure which further give rise to the embryo and embryo will form the new individual. Second male gamete, इस process को हमने क्या बोला? Syngamy. याद रखना है, this process is called syngamy. Fusion of male and female gamete. Syngamy हो गया. Second fusion क्या था? Male gamete is fusing with 2 polar nuclei and forming a endosperm. This is called triple fusion because 3 nuclei are fusing here. So this is called triple fusion. So you can see that 2 fusions are taking place in the same embryo sac. One is syngamy and another is triple fusion. So collectively this triple fusion and syngamy is are known as double fertilization. So question आता है, why it is called double fertilization? It is called double fertilization because in a same embryo sac the fusion is taking place twice. One syngamy and another triple fusion. That is why it is known as double fertilization. Now let us look at the fate of some floral parts after fertilization. Petals, stamen, style and stigma they wither off, गिर जाते हैं वो. Calyx में either fall off or may remain intact. कुछ केसेस में इंटैक्ट रहता है कैलेक्स, जैसे कि ब्रिंजल के केस में. The ovary enlarges and forms a fruit. और ओव्यूल्स क्या बनाते हैं? सीड. तो यहाँ से देखें, ओवरी कन्वर्ट्स इनटू फ्रूट. ओवरी वॉल फ्रूट वॉल में कन्वर्ट हो जाती है, दैट इज कॉल्ड पेरिकार्प. ओव्यूल्स फॉर्म सीड. प्लेसेंटा फॉर्म्स द स्टाक ऑफ सीड. जो इंटेगुमेंट्स थे दोनों, तो आउटर इंटेगुमेंट बनाता है टेस्टा, इनर इंटेगुमेंट बनाता है टेगमेन. टेस्टा एंड टेगमेन आर सीड कोड्स. सेकेंडरी न्यूक्लियस कन्वर्ट्स इनटू एंडोस्पर्म. एग सेल और सिनर्जिट्स ये एम्ब्रियो बनाते हैं, एंड एंटीपोडल सेल दे डिसऑर्गेनाइज्ड.